Hello and welcome to session 29. Today we will be discussing a very small topic, porphyrin, uh, in which we'll be looking into the properties of porphyrin with one example. Porphyrins are any class of water-soluble nitrogenous biological pigments, derivatives of which include the hemoproteins. Porphyrins combine with metals and proteins. The examples of hemoproteins are the green photosynthetic chlorophylls of higher plants, the hemoglobins in the blood of the many animals, the cytochromes, enzymes that occur in minute quantities in most cells and are involved in metabolism or oxidative processes and catalase, and also widely distributed enzyme that accelerates the breakdown of hydrogen peroxide. The structure of porphyrin. Porphyrins are cyclic molecules formed by four pyrrole tetrapyrrole rings linked by methynyl bridges. Different porphyrins vary in the nature of side chains that are attached to each of the four pyrrole rings. Each protoporphyrin contains vinyl methylpropionate. The distribution of the chains may vary, and we will see the structure here. Uh, Porphyrins are cyclic compounds that bind metal ions, usually Fe2 plus or Fe3 plus. So that is porphyrin plus metal is equal to metalloporphyrin. Uh, Most prevalent metalloporphyrins in humans is Heme. Metal here is iron. Heme consists of one ferrous ion Fe2 plus. In the center, protoporphyrin 9, a tetrapyrrole ring. Heme is the prosthetic group of hemoglobin, myoglobin, and cytochromes, catalase, peroxidase, NOS, and tryptophan pyrrolase, and so on. So heme plus globin protein is equal to hemoglobin. So porphyrins are organic pigments of both natural and synthetic origin, uh, all of which contain the porphyrin ring. In addition, porphyrin chemistry deals with various analogues and derivatives of porphyrins, and particularly with their metal complexes. The porphyrin molecule is usually described as a union of four pyrrole rings linked by methine bridges to form a macrocycle. The properties of porphyrins are strongly colored compounds. The variety of available colors is apparently limited, although there is an observable inclination towards red and purple. The color is a consequence of the complicated electronic spectra of porphyrins, which contain intense absorptions in the visible region called Q bands. So hence we sometimes call cytochrome Q. Uh, Even more intense, 10 times and more, is the Soret band found in the near UV, so named after its discoverer, Soret. So this is the typical structure of a porphyrin, and these are the four. These are the four tetra pyrrole rings, and linked by the methylene bridges, 8 side chains, two on each pyrrole. So these are the side chains, two on each pyrroles, and these side chains may be acetyl, propionyl, methyl, or vinyl groups. Different porphyrins vary in nature of side chains depending on substitute groups divided into 1 series and 3 series. 1 series symmetrical arrangement, 3 series asymmetrical arrangement. Yes. So this is the overview of hemoglobin structure. In the center you can see the ion Fe, Fe2 plus, and heme is comprised of a porphyrin ring structure with one iron atom chelated in the center by four nitrogen atoms. This is the site of a reversible oxygen attachment. Each hemoglobin molecule contains a heme group attached to each of the four globin chains. Four total heme or molecule which is capable of carrying up to 4 molecules of oxygen at one time. So this is the structure of hemoglobin. So in the center we are having a metal ion which is a Fe, and it is connected by four nitrogen atoms, and we have four pyrrole rings, and to which the side chains are attached, and one hemoglobin can carry up to 4 oxygen molecules at a time. And these are the references that I have used. So thank you one and all.